Okay. First of all, thank you for the invitation to talk about this project. Mainly, I think we have been doing before multiple presentations about pride. As I said, my name is Yasset Perez-Riverol. I am a project leader of pride resources at EBI. And today I will present the latest results we have in one project called Proteogenomics, Pride Proteogenomics, which is basically trying to represent proteomics information into Ensembl and other genome browsers. Please, if you have any questions, you can basically type it in the box. The main idea with this project is being able to take proteomics data, which mainly is produced by laboratories. Not within most of the data that we have in PRIDE is not in the context of a proteomics a genomics experiment. We would like
interruption. Your screen is not currently being shared.
Yeah, it's shared, I think. Okay, let me check again. Sorry. Oh, sorry. Okay. Sorry, can you see it right now?
Yes, perfect.
Okay, sorry about that. Yeah. Then the main idea with this project is to being able to represent, as I said, proteomics data into first Ensembl browser, but also in other browsers. First of all, I would like to say how the data flow into the PRIDE database. PRIDE is the proteomics identification database, which is mainly stored in the EBI, and is actually one of the main databases to store mass spec evidence, and it is one of the members of Proteome Exchange with other teams and other databases like Massive or Peptide Atlas. Wherever a data set, proteomics data set, is produced by one laboratory, these data sets is actually submitted into PRIDE using our PX submission tool. We have two types of submissions. In one submission called partial submission, we only have the raw data, but in the other submission, which are called complete submission, we have also the list of peptides evidence and product that has been found. The idea of this project is actually take all this peptide evidence and try to accurately actually try to see which are the more relevant or more reliable peptide evidence, and try to map those peptide evidence into genome coordinates. And after that, for each individual project, then we generate a Trackhub that is actually submitted or registered into a Trackhub registry. I will explain a little bit what is a Trackhub registry. And then this can be seen and searched when you are in the Ensembl on UCSC browser, and you're looking for proteins evidence or for proteomics evidence, then you can search for these data sets and then see the results. This pipeline we have built have multiple components. First, all the data that is deposited in PRIDE, all this peptide evidence coming in MC-IDML, MC-TAP, and PRIDE-XML. Not all of those peptides are relevant, basically. You need to assess the quality of each peptide. And for that, we actually use, we have developed a pipeline based on a tool that we developed like 2 years ago and it's published in JPR, called it PIA, is the Proteomics Inference Engine, that enable us to filter all peptide evidence at 1% FDR and then at peptide level 1% FDR. And then we apply a couple of filters, like for example, that each peptide needs to have more than a amino acid, and this is basically aligned with some guideline from the HPP project, the Human Proteome Project. And at the end, you have a file like this. We call this file like PoGo file. When we have the URL of the PRIDE dataset assay, we have the peptide, and actually we can also expose some information about the quantitation. In this case, is we actually expose the spectral counting for each peptide on each assay. There is one relevant thing here, and I just want to give the idea that we also kept also the modifications. Yeah, is every modification that is represented on one of these files is actually translated into one biological relevant modification. Let's say if you have oxidation, we actually put the oxidation in the correct place in the peptide, and so on and so forth. Okay. When this is done, then we use a tool that we have developed in collaboration. We have been developing collaboration with the Sanger team that actually tried to get those peptides, and using the protein sequence databases generated by Ensembl and the GTF file, we actually are able to map all these peptides evidence into genome coordinates. Okay, and then at the end we have a bed file, and this bed file contains for each peptide evidence where is the position in the genome of each peptide. Okay, I need to say, and this is I will go a little bit back, that we do that systematically for every submission. Every time a submission arrives to PRIDE, if it's a complete submission, automatically our pipeline pick that submission, assess the quality of each peptide evidence, generates the bed file, and then register these bed files into the Trackhub registry. And finally, we send back an email to the submitter saying, okay, your Trackhub has been generated and is published in this URL, and it can be used by the community. Okay. At the moment, only for human, this presentation will be mainly focused on the architecture, all the tools, and the resource. But we already are in some way proud about how many evidence we are actually getting. We are doing some basic statistic about the number of peptides. For example, these are the number of peptiforms we have already mapped into Ensembl, around 4 million peptiforms only for human. And this is actually the distribution around by chromosome, okay? And these are actually the number of peptide evidence modified, okay? And then for modification, we consider only, I think, 10 modification, the more biological relevant, like phosphorylation, oxidation. We also have acetylation, and so on and so forth. Everything can be seen in our GitHub page, or you can contact me if you are interested in doing data analysis on top of this data. For example, we also one of the important thing of this project is that because we can do this in high throughput, systematically for every submission, we know that in PRIDE we have a lot of submission coming from multiple species, yeah. And then we can do also this for mouse, and these are the first statistic about mouse. And these are the number of peptides and number of modified peptides. Okay. But the most important thing with this resource is that we want to, rather than ourselves analyze this data and so on, we want to put this resource available for us. Okay. Then when we have the PoGo file, we have a tool that we have developed. It's a framework that we have developed in Python that actually enabled to publish the Trackhub. Okay, and then a Trackhub is basically some minor information about where this data set come from. The URL actually of the big, in this case, big bed file, because big bed is a binary file that enabled a fast transfer of the data and so on. And then these Trackhub are published automatically in the Trackhub registry. And the Trackhub registry not only contain data around proteomics, but also transcriptomic, genomic variants, and so on and so forth. Okay, all of these data sets can be searched in the Trackhub registry, and then can be visualized in whatever genome browser the user prefer. There is a really nice work, as I said, within this Trackhub creator tool, and this is actually a work that has been led by Manuel, one of our developers. And there is a couple of things, interesting technical stuff here. For example, the Trackhub creator automatically download all the Ensembl FASTA file, GTS file, automatically also convert the input from peptide identifications into a bed file, and also convert this into the big bed, but also do a fancy thing, for example, like make all the Trackhub compatible with Ensembl genome browser and UC genome browser, which is actually one of the challenge here. I mean, it's basically all the data we provide, we want to be compatible as much as possible with the tools that are out there. Okay. How a Trackhub looks. When you go to the Trackhub registry, it's a simple registry that has the search capabilities and so on. And then you can actually search for a data set. This is the URL, trackhubregistry.org. And then you can actually search by, for example, I don't know, cancer proteomics, yeah? And then you can see here, for example, around the first thing is that we actually highlight the original title for the data set. We're doing this at the level of each data set submitted to PRI. And the important feature here is to give to the original author of the data set credit for putting this data available, yeah? And these are more functionalities that the resource and PRI team and PRI resource is given to the community, but the credit is actually for the authors of the data, yeah? Then when you have some title around the data set, some description about the data set, you have the species and actually the assembly that was used to map the peptides into the genome coordinates. And finally, you can select here which genome browser do you want to use, okay? There is a lot of work in the middle. We actually, for example, whatever arrives to PRI, it can be represented in multiple PTMs representation. You can use Unimod, PSI mod, and then we map all of those into a canonical, let's say in some way, representation to being able to represent all these data. At the moment, we are supporting 19 taxonomies, and we are trying to get more data around that. How the data will look at the end in a genome browser. This is, for example, an example with UCSC, and you can see here all the peptides that has been found, and then if you click the peptide, then you can actually see some information about from which data set it come from, and so on and so forth. Okay. We are also the capabilities that the browser has right nowadays to show quantitative information is not enough. But we know, for example, the IGB or IBG browser is actually able to show some information about quantitation. We are moving also in this direction. However, the data in PRI is, until now, have been showing what we do with independent experiments, yeah? But a lot of users in PRI are interested to see how much evidence across whole PRI is actually can be mapped or can be seen in the genome browser, yeah? And this is basically a unique view across all the peptides evidence within across all PRI that should be mapped into that. The amount of data that PRI has, this is a statistic from last year, is growing every year. And to condense all this data set and peptide evidence, we published in 2016 the PRI cluster approach, which is actually going back to each spectra that are submitted to PRI, and we cluster all together, and then with that we can assess across the entire resource, we can assess the quality of each peptide evidence, yeah? Then we can say, with some p-values of score, we can say this peptide is relevant across the entire resource. This the way this is implemented, this has been published in Nature method, is we receive all these independent submissions. These submissions are sent to an algorithm that has been implemented using big data Hadoop technology. And then we generate a peptide table when we actually say for this peptide, this is the amount of time that this peptide has been seen in the whole resource. Just to give an idea, and this is actually some data from the publication in 2016, the amount of peptide P4 proteins that can be represented by peptides in pride cluster is actually quite high. You know, it's like we have a lot of peptide evidence in pride that actually coming from pride cluster that actually can be used for like evidence for uniprot proteins that do not have any mass spec evidence. All these cluster data, all these peptides are also mapped for each individual species into genome coordinates, and every species has a pride cluster latest release Trackhub into the Trackhub registry. Yeah, and then you can see here, for example, this is for example for cow, ratus, and human and mouse, all the pride cluster data. We mainly what we aim with this resource is to provide data to others. Yeah. But we would like to be interested also to see how much data we are providing at the value of this data. And this, for example, this is the coverage. We have developed a package in R called PepBed that allow users, for example, to download all the data that we put outside, and then, for example, do simple statistics about, for example, chromosome coverage that we have within Pride. Within all the experiment, and also you can do really nice visualization about, for example, the coverage for each chromosome, but take into account the modification. These are the unmodified peptides, and you can see here the amount of phosphorylations in red, phosphorylation peptides that we can cover. It's interesting the coverage that we have actually for human and mouse, almost for every single chromosome we have more than 40% of coverage, but we are moving forward to put more data. We'll explain a little bit about that. Okay, what we are doing also within this package is actually, because we have data across multiple experiments, we can actually assess the significance or the probability that one particular phosphorylation or oxidation, for example, these two modifications, are significant across the whole genome. And now we are generating a statistic about how significant is, taking into account all this information, how significant is, these plots are only for human, is how significant are some specific phosphorylation sites and oxidation site for the whole entire human genome, taking into account all the experiments that we have processed. As part of this project, we are also working. We have been working and has been published last year in a new version of PepBed file. As you know, PepBed file is mainly a file format for representing biological information into genome coordinates, but it was important for us to have a file format that actually represent mass spec information. For example, the PSM score, the reference to the PSMs, for example, quantitative information, and so on. You can read more about this. It was, as I said, just published in Genome Biology, and we are now trying to export all of our data into ProBed to be consumed by our users. What is next? Yeah, as you can see, all the data that I was showing, I mean, is we are happy that this pipeline, now that is in place, will increase the number of evidence wherever data become more public in Pride. Yeah, and then if user, and this is actually really important, submit more complete submissions into Pride, not partial submission, then more data will become public in this resource, in this proteogenomics resource. Then that is actually a way of promoting that the proteomics community actually do more complete submission. That means that more software like MassQuant and other software needs to export their data results into standard file formats. But knowing that the amount of data we have in Pride, we would like to start reanalyzing data, and then we have starting to do that with different pipelines using MassQuant and also SearchGUI to actually increase the amount of evidence we have in this project. Just to give you an idea, for example, only in human, Pride actually now contain almost 1,000 data set. Also in mouse, we have almost around 300 data set, and so on and so forth. This is everything I would like to present. I would like to thanks to all the people that have been actively involved in this project, especially to Manuel Bernal Linares, who was the actual developer of the Trackhub creator platform. Christopher from the Sanger team has been working in the PoGo tool, and Tobias has been working with me in all the pipelines, Johannes in the cluster, and the two PI of this project, Jyoti from the Sanger Institute, and Juan Antonio Vizcaino, who is the PI of our team. Thanks, and questions.